Okay, welcome everybody. Glad to see a great turnout. And this is basically the kickoff of our DSS series. So for those that were here before, this will be repetition. For those that don't know the series, that are new on campus or just starting as graduate students, this is basically the CCMA Distinguished Seminar Series that was founded by the CE department, jointly with the support of Chiang Mei, who was a faculty here in CE and also in MECI. And so the theme of the series is to promote diversity in research and bring great speakers from around the world that really cover all sorts of interests that span the CE department. And so, for example, this term, we're going to have 3 speakers. So today's speaker, Dr. Amaral, who will be introduced by my co-host, Sergey, in a moment. And then next week, we will have a series, a talk that will really focus on interfacial flows and fluid dynamics by Dr. Israelovici. And then the week after that, so they're back-to-back this term, which, so you have to stay intense and tuned in 1 month. All come in all the Mondays that will follow each other. But the last one of the term will be with Dr. Weller, who will be talking about transportation and networks in that domain. So 3 very different domains, but that will basically span all sorts of areas in the department. And next term, if you're curious, you can already see the list of speakers. They are listed on the website. And so you can already stay tuned and mark your calendar. So for today's talk, I will leave it to my colleague Sergey to introduce Professor Amaral from Northwestern that will cover basically complex systems.
Well, it's my great pleasure to introduce Luis Amaral. Luis was born and raised in Portugal, Lisboa, and he got his PhD degree in physics from a place not far from here, so Boston University, under the the supervision of Gene Stanley, one of the most prolific physicists I know. And from there, he started basically developing a very successful multidisciplinary research line. After that, he went to different postdoctoral fellowships. MIT was one of those places, Boston University, Harvard Medical School. And after that, he went to Northwestern, where it's not surprising that he's now professor of physics and astronomy, professor of medicine, professor of chemical and biological engineering. And that basically talks about the diversity of Luis' work, which has been very influential and has been doing great contribution in many different fields, from social sciences, economics, systems biology, developing the functional cartography method to understand the processes underlying metabolic protein networks. And without further ado, I just leave Luis Amaral with you guys. So.
It's really a pleasure for me to be here talking to you. Thank you for the really nice introduction. I try to be very casual, so please feel free to interrupt anytime during the talk for clarifications, for arguments, for whatever. I have way too much material, so at some point I might ask you, should I do this or should I do the next one, and just skip one point in the story. I decided to go with this title because, we have a presidential election coming out and all this stuff about immigrants, and I said, why not? Actually, I was noticing as Lydia was talking that I was just behind her, and I started to have a Trump moment. I was like, oh, no way, make sure that I have a certain distance so I'm not kind of appearing creepy and stuff. So, what am I talking about? I've always been very interested in research from many different areas, but I actually started as a physicist, which, according to the Saturday morning breakfast cereal, it's pointed by the fact that you don't very much pay attention to some small differences, right? All cows are circles, and all numbers are 10, 100, or 1,000, and nothing else matters. But more seriously, one of the things that really attracted me to physics was the scale of it. So even though I probably cannot call myself a physicist because I don't do anything that would be remotely similar to what a physical system is, I still am very influenced by this. And if you have not seen this movie about Power of 10 by two of the greatest designers of the past century, Charles and Ray Eames, you should. And I can give you a description. Essentially, the way that the movie starts, it starts with this couple having a picnic in Chicago in Millennium Park. And it started showing them at about a scale of 1 meter. And then the movie actually goes in two directions. First, it starts going down at scales that are smaller by factors of 10, and you know, kind of looking at the level of skin, cells, DNA, particles inside the atoms, and all of this stuff. And it's, as you zoom in, the length scales and the time scales for a process to occur are shrinking fantastically. But then it actually accelerates, goes back, and then it goes and starts looking at things at bigger and bigger scales, right? Looking at them from 10 meters, looking at the entire park, looking at the entire city, the entire planet, the entire universe. And then this is the appealing thing about physics, is that you are not focused on a single scale. You are not focusing on phenomena that occur at a given time scale either. It's not all about seconds. It can be something that are billions of years. It can be something that is a femtosecond. So it really is exciting that you as a human can actually think about phenomena that occur in such wide range of scales, both temporal and spatial. And I think that interest kind of motivated me to be interested in problems in other areas that usually would not fall under what would be called physical phenomena. And so one of the areas that, and I'm going to tell you about 3 vignettes of some of the research that's going on in my lab about what we did and what were the problems that we were trying to address in there. And the first one is related to my interest in understanding creativity. What are the conditions that foster creative thought and productivity and innovation? And how do innovation spread among people? And you know, we have done work trying to estimate the significance of creative works, and this is not as simple as it would appear. Just to give you an idea, if you think about the citations of papers, probably the most cited paper right now is an essay in molecular biology. It's about 300,000 citations. Okay, just to give you also an idea, George Whitesides, who's a very, very famous chemist at Harvard, has a total of about 90,000 citations from all of his papers. Right? This is one of the most famous chemists alive. And all his works together, the citations of all of them, is about a third of the most cited paper. Right? So how do you decide? Is every author of that paper so much better than George Whitesides? Because one paper of them, right? Or you can think about J.K. Rowling, right? Is Harry Potter billions of times better than, you know, the guy that had the Nobel Prize for poetry because his book maybe sold 1,000 copies? It's hard, right? It's hard to quantify the impact of something and the significance. And so we looked at a case in which you actually could do something, which was movies. But I've also been interested in how people create significant work. And one of the things that is going on right now is that teamwork is seen as something that contributes to creating innovative work. And lots of science right now are being done by teams. And you could ask, are there some characteristics of teams that are more related to good work being done? Right? And we did a study of that in which we found that there were some characteristics of successful teams in terms of producing more creative work. And one of the things is sort of the idea of new blood, of making collaborations with other people that are also experts, but are not experts exactly in the same area as you. But one of the things that we couldn't do in any of these pieces of work was figure out what is it about how good teams work. And if you look at papers or if you look at movies, you see the product at the end. And you can say, is this great or is this crappy? But you actually don't see how people interacted during the process. And that is another piece. Are there something about how people interact during a project that is a sign of that something good is being going on? And I'm lazy and I'm cheap, so I the traditional way to do this is to have someone going and shadowing a team at work and taking notes and stuff like that. And I thought if I had to do that myself, I think I would prefer to shoot myself than do it. So I don't want to put anyone through that. Or you'd pay someone, but then who has the money for that? So I thought, how can I get information about that? Oh, and then I thought, I love soccer. And soccer is a place where you can see a team playing and their interactions, and you could figure out, oh, this is the way in which good teams of soccer players interact tells you about how high their performance was or not. But even that was not so good. First, because most people tell you, I mean, looking at soccer is not a respectable activity for a scientist, right? So right away, it's kind of, okay, I have to move away from that. So let's look at something that is more respectable. And so I started talking about where is this place in which I can look at teams working and where I can get good information. So, you know, one situation that, oh, and the solution, of course, would be get a bunch of undergraduates in a computer lab and do some experiment. And I thought, nah. First, you know, it's really not life and death. No offense to the undergraduates, but they are most of the time still not highly qualified at anything. I hope there are no undergraduates in here. Okay. Except for the undergraduate present. So I wanted to have something that kind of gave you good information about teams like doctors, lawyers, architects, scientists, in which you have a team of people that have very high qualifications, that are highly capable, they are working together on a project, and the decisions that they make matter. And I also want a case in which the outcomes are clear relatively quickly. That already said architecture is not a good one. So medicine, right? If you make a mistake, your patient can die, so that makes it sound like life or death. If you look at people in the ICU, the chance of them dying is pretty high, so you find out if they screw up quickly, right? Within a few weeks, you found out if the person died or not. Sorry that I'm looking so upbeat about people dying in the ICU. It's just because it's 5 or something. And you have highly qualified people, right? You really have people that have a bunch of skills, so it's not trivial, right? It's not like the guy that was operating the train and went 3 times or 10 times as fast as the speed limit and crashed, right? That's not the kind of mistakes that we are talking about in medicine. So we want to understand medicine, and you can look at the leading causes of death, and you know the usual suspects come in. But actually, there was a report that showed that lots and lots of patients die because of medical mistakes. Either the doctor did the wrong thing, or the doctor forgot to do something that was important and the patient died. Okay, so to give you just the numbers, the later studies said that about 200,000 people a year die in the US at hospitals because of medical mistakes. You put that on a rate per 100,000 and you see that medical errors are the 3rd leading cause of death in the US. And if we could address medical mistakes, we could save a ton of lives without a miracle drug, right? So this could be something in which it would be a good system to study. So you can ask, why do errors happen in medicine? And the reason why they happen is because things are damn complicated. So this is a patient in the ICU. You can see that there are many machines around recording stuff. These patients are very, very sick. There is also a very large team of individuals treating them. Not only that, the individuals being part of the team change quite dramatically, right? So you have lots of people, lots of information, lots of decisions that need to be made. So you have lots of opportunity for mistakes to occur. I cannot read someone's handwriting, or I forgot to tell this piece of information to the physician that is going to replace me, right? So we could look at this as a way to find out where are mistakes more likely to happen, what could we do to address them. So what are the behaviors that need to change, and how can we change behavior? And this was done in collaboration with a colleague and friend, Kurt Weiss, who's an MD. And I think one of the things when you try to go into someone's field that it always turns out to be really, really good piece of advice is to find someone in that field to collaborate. Because you avoid making really stupid mistakes, and you avoid looking at problems that no one in the field gives a damn about. So if you are entering there illegally, try to find an accomplice to hide you in their house. And also a colleague who's also a physicist was working on this. So one of the things that people have been thinking in medicine is that if we could make use of resources like checklists, we could avoid mistakes, because these have been shown in a number of contexts to be really, really helpful. And in fact, there was a book by Atul Gawande, who's also a wonderful writer, and he writes a lot in the New Yorker, and he wrote the checklist manifesto, which was about how checklists could really improve outcomes. And they really improved outcomes in medicine, right? I mean, we all heard about the idea that, you know, the patient is supposed to be writing in their legs, this is not the leg you should operate in the other one, and actually probably writing in their liver, no, do not remove my liver, the liver is not the problem, it's the knee. So all of this stuff, and it really has an effect. And it has an effect also in things like not forgetting sponges inside the patient, not forgetting scissors, not forgetting knives, you know, things that you would imagine would not happen, but you know, you are doing something, suddenly you cut a blood vessel, there is blood everywhere, you are trying to fix that. In the meantime, you forgot that a pair of scissors fell inside the patient, until the following day when the patient is in a lot of pain and you take an x-ray and you find out, oh, scissor. So the reason why I believe it worked so well in surgery is that you find that you made a mistake really, really fast. So right away, the following day, you said, I ignored the checklist, and here you are. Pair of scissors inside the patient. So you realize, oh, okay, if I use the checklist, it works. But there are other contexts in which it doesn't work. And so this is the checklist that was developed at Northwestern Memorial for the intensive care unit. And the form looked like this, had bunches of things, some for the physician, some for the pharmacist, some for the nurse. And actually, most of the forms, after they had been given to a patient, looked just like this, except that they had the patient name. And the reason why was that it was very hard to figure out that it had any effect. And the physicians really thought, this is actually distracting from my workflow. And who the heck would forget to look at these things, right? I mean, these was things that were covered in Medicine 101. Do I really need a checklist to check that out? Well, so we made an experiment. The ICU, the medical ICU, had two teams. Each team was assigned a separate set of patients. So we actually could have one team that was a control, another team that was the intervention. And the intervention was very simple. One of the team members, actually, unbeknownst to anyone, was supposed to be looking at the checklist items. And if, you know, for those of you that are not familiar, that in the morning the team goes and does rounds, and during rounds they discuss each of the patients. And when they are discussing the patient, they were supposed to be looking at the checklist and see, well, have we addressed these or not? And what this person did was, if there was an item in the checklist that was not addressed during the discussion of the patient, they would bring it up. Oh, what about weaning the patient from the ventilator? Nothing else, just asking, okay? Not saying, oh, we need to do this, or the checklist tells us that we should do this. So it's very soft intervention, just nudging. And so you can ask, did this make any difference, okay? So we had pre-intervention data. We had data on 1,200, almost 1,300 patients. Then we had the two groups. In the prompted group, we had 125 patients. In the other one, we had 140. And if you are at a top hospital in the US and you are admitted to the ICU in a similar, in a ICU of an hospital with a similar population, the chance of you dying is about 21%, so 1 in 5, okay? For the control, what we found out is that this was the prediction for the control. So you actually have a score based on the signals, on the signs, physiological signs of the patient about what's the likelihood of the patient dying. And so you could estimate that for the control population, that probability had been close to 22%. The observed was essentially 21%. So very well matched. This was for the prompted. The probability estimated was 22%. The observed was 10%. A 50% decrease in mortality rate. So instead of you having 1 in 5, you had 1 in 10 chance of dying by having a little nudge about the items in the checklist. So clearly, this looks too good to be true, right? So let's make sure that these make some sense. Now, what you can do, you can look at the patients and getting their vitals, you can predict what their mortality is going on. And so if we think about it, errors, the effect of errors, we can think about two populations in which errors are not very serious. One is the population of not very sick ICU patients, the ones that are going to be discharged after a couple of days. Then you have the ones that are very, very sick, and it's going to be a miracle if they make it anyway. Okay? For those two populations, mistakes should not be a serious effect. The first one, staying there too short a period of time for a mistake to occur, likely. The other one, well, I mean, the situation is so bad, your mistake is not going to make it that much worse. It's the ones that are kind of intermediate degree of severity that you'd expect the worst outcomes. And this is what you find, that these are the two groups. These two intermediate ranges are the ones where you find the biggest reduction in mortality, right? The ones that stay long enough for a mistake to occur. And a mistake, typically something like someone forgot to change something and now you got an infection. You got an infection at the ICU in an hospital. Think about that. What's the chances that that is a relatively nice bug? The other thing is that if you look at how long the patients stay in the ICU, you see that until about 72 hours, there is no difference. 72 hours is the time that is necessary for the probability of you getting an infection on a line that has not been changed becoming essentially 1. Right? You stay long enough past this, you get an infection, and then your length of stay becomes much, much longer. Not only that, your chance of dying becomes much, much bigger. So both of those results tell us that this is not likely wishful thinking. That this happens in the populations that we expect it to happen. And it tells us what dramatic impact these mistakes have on outcomes. Really, really tremendous. Now, nothing has changed at Northwestern Memorial. They decided that the paper checklist did not work, so they moved towards an electronic checklist. The electronic checklist is even worse. It actually requires much more disruption of the workflow of the physician. So it's not being used. Now you may think 20% and 10% is a very big difference in mortality rate. The attendings stay in the unit 1 week at a time. They really have trouble seeing the consequences of the decisions that they make on the patients. And we are still talking about small numbers of people dying. So it's actually that it's very hard for them to detect that there's been a change. So this is a case in which, you know, something that has a big impact but has not been implemented in a way that works with workflow is not being adopted, and it's costing a huge number of lives and is costing a huge amount of money. Every night that you stay in the ICU is probably another $3,000. So just look, the average length of stay is almost double. We are spending a ton of money. So I have 2 more stories. The next one is relatively social behavioral. The last one is more biological. And I don't know which one you guys would prefer. This one? Okay. The one person to vote as make the decision. This is elections in the US. The one person in Ohio that is making the difference.
I have actually a comment or a question about the previous story.
Yeah.
So both my parents are physicians, and I've heard many, many rants when I've been home about how inconvenient and annoying electronic medical record systems are. Have you thought about any of that work, which is, I guess, not directly related to EMRs, but it's certainly a similar story of something that's disruptive or not convenient for the workflow. And
Do I have 2 hours to go on a rant about that? I mean, I have two hypotheses. The people that design interfaces for electronic health records are either sadists or completely incompetent, because you cannot believe what the electronic checklist looks like. Okay, yeah, I'll stop. I have to keep my blood pressure low and stuff like that. Yeah. No, but this is really the problem. It's one of the biggest problems, is that people have no consideration for, and then they say, you have to do this. But it's like, I have a million other things, and you haven't shown me that this is actually helpful and stuff. And we are doing something about that, but that's another story. So,
I've never worked in a hospital, but
neither did I.
Okay.
And I avoid them like the plague. It's like,
how can it be so hard to get doctors to use a system that you've just demonstrated works, right? I mean, sure, maybe they don't see an individual patient, but you can take some examples from your study and say, look, then the hospital administration can say, look, we're going to save $1 gazillion if you do this.
and the hospital will tell you, so the healthcare, okay. Everybody knows that if you hired more nurses, things would be much, much better. But hospitals do not hire more nurses. You all know that if you had sort of more continuity of care, outcomes would be better. But that is, so the system is insane. And we all know how easy it is to change the healthcare system in the US. So my approach is trying to see what can I do that would improve the situation, but actually would work with the current system without hoping that a miracle occurs and things change. I mean, you would say, oh, you could get someone to do this prompting, right? Because then it would not be disruptive of workflows at all. When I made that suggestion, I mean, I was laughed out of the room. The hospital would actually hire more people. Actually, the two hospitals the two big hospitals all the big hospitals in Chicago had an agreement. They were fine because they had made an agreement not to hire away nurses from one another, because that would increase their salaries. So, Okay, let's talk about something better, and you will see. So I was also interested in human activity, and we did some stuff related to communication, and we found that there were some really robust patterns about how people communicated through email and letter writing and all of that stuff. But there are other types of actions that are human activity that we could try to be understanding from a statistical perspective and finding out what's going on. And this is work done with my colleague John Hagen and Adam Pa, who used to be in my lab. And we looked at something that, let's move from people dying in the ICU to something even nicer, murder. And so this is the data about the murder rate in the US. And you can see that there are these very non-constant behavior, right? This is not fluctuations around a value, and these values are horrific. Just to give you an idea of how horrific they are, this is the data for Canada, and that's the data for England, and it gives you an idea about how horrific we are talking about. But of course, this was not horrific enough for us, so we looked at school shootings instead. So when you look at school shootings, there were a bunch of databases that were collected, and one of the issues that we found with all of these data was that they had been collected according to very different criteria. One of the biggest difference was what they meant by a school shooting. So some of them defined a school shooting is anything that occurs within a given distance of a school. And the given distance could be several blocks and things like that. And so, that distance could have a very different meaning depending on you are. If you are on a less densely populated area, 500 meters might be much less than a block in a very densely populated urban area. So we thought we really need to restrict this stuff. So we went through all the records in all the databases, compared them across original sources of what had happened. We made sure that these events involved either as perpetrator or victim, either a student or a staff member or a teacher in the school that occurred on the school campus, and all of this stuff. And we came up with this, what we called our consensus database that had 382 events. And you can see that we didn't look at data before 1990 because even the 1990s kind of, once there were these big, significant mass shootings at schools, people started collecting these data much more carefully. Also, it started to be recorded in the internet and publicized nationwide, so data became, we didn't need to worry about maybe some event occurring that hadn't been captured anywhere. So we had much higher reliability. But what's interesting is that you can see that there is kind of a very striking pattern. This is clearly not uniform. And so this is the data actually with the number of fatalities that occurred. Green here is 0. Yellow is between 1 and 3, and red is more than 3 people dying. And you can do a fit of this to a Poisson process with some rate, and you can see that you can actually fit some constant rate over some periods. And you could figure out how many different pieces you break these, and this was sort of the best description. The idea that there are some changes in rate. And we were looking at explanations for these, or at least things that correlated. And you could look at things like gun ownership. And what you find out is that the percentage of families or households in the US that own guns has actually been decreasing over time, which is funny because you always think about the number of people that have guns and that have lots of guns is becoming smaller and smaller. Then the fraction of people that do not own guns has been growing easily since the 60s. So that was not an explanation. And then we thought, well, maybe these shootings are a reaction to something in people's lives, right? Something is making them feel upset, angry, and that is coming out through anger and violence. And just because guns are pretty easy to get in the US, you actually can express your anger and unhappiness by getting a gun and shooting someone. So what we did is we looked at the data and compare it with an employment rate. And if you look at that, you see, oh, employment was high here, was lower here, was higher there. And there seems to be some correlation in there. And that is for the national level, but you could repeat the same analysis at the regional level, in which now this is the unemployment rate for the Northeast and for the Southeast and the Great Lakes and the Pacific. The other regions are too small in terms of population, and you get so few events that there is hardly anything that you can do. We can also do it for individual cities. Again, the number of cities over which over this period there were enough shootings for you to be able to do that analysis are only 6, fortunately. But again, you can see that there is some occurrence about more likely when the unemployment is high. And you can kind of make this analysis more precise by saying, okay, if I normalize the unemployment rate for the period between 0 and 1, okay, in each aggregation level that I'm considering, and then I can say for a given month, what's the probability conditional on there being a shooting or not of the unemployment rate? And at the national level, you can see is that when there are more than one event, you can see that that in blue tends to occur more in months where the normalized employment rate is higher. The same at the regional level and the same at the city level. So it's really that there are more shootings when unemployment is higher. And all of these things, if you do the KS test, is 10 to the minus 3 p-value. So it really suggests that what's going on here is a reaction to economic insecurity and distress, is that people are feeling upset. They are feeling upset not only about what's going on in their homes because their parents are maybe getting unemployed or facing harder economic situation, but also that if you are about to graduate, typically you would think, well, I went through school, I delayed the opportunity to maybe get a job and make some money because I'm expecting that at the end I will be able to get the job that will enable me to have a decent life. And if you see that there is no hope of that, it's natural that you get angry. And so what John did in putting these results in context is really about what this is telling us is the breakdown of the school to work transition. As people see that there are less options when they graduate, they see that all this effort that they are put in of following the rules and doing what was expected has led to nothing. And one of the things that we actually found out is that for the second period in which there are more shootings, it's when, right, there were two kind of periods of heightened rate. One was around the early 90s, and the other one was associated with the Great Recession. And what we found out with the period for the Great Recession is that that's where there was a dramatic increase in the shootings at colleges. And this was the issue that it was not only now people with high school degrees that were facing difficulties of finding jobs. In the last recession, even people with college degrees or associate degrees started to having problems finding jobs. So it's really about this anger at the breakdown of this transition. So it's not like that this gun violence is about people that have mental problems. Well, it is also about that with some people and with the mass shootings, but it's really a reflection of economic insecurity. And a reaction and a display of anger at something of feeling their expectations kind of burst. Okay, so I can promise you that this one, well, actually I shouldn't promise anything. At least there will be no death or murder. So another area in which I'm very interested is related to processes that occur at the molecular and cellular level. Sorry. As Sergey said, we looked at the structure of metabolic networks and did a lot of work around trying to organize that data. But I've also been interested, to me, the cellular level is a stepping stone to understand what happens at the level of the entire organism and more of an integrated view of the system. And here, what we were interested in understanding is why do biological systems have the characteristics that they have? And so when you look at biological systems, especially during very sensitive processes, like, for instance, development, in which you start from cells that have the potential to be pretty much anything, and now they are making choices about what type of cells they are becoming. If you screw this up, and we can actually do that artificially on organisms, right? If we use fruit flies, we can make them grow legs instead of antennas and things like that. So it's a very, very sensitive process. in which mistakes can be having very serious implications, is also a system in which if you have faster dynamics occurring, then it gives you an advantage. Development is a very sensitive time in the organism's life, right? If you are a butterfly, you are inside the cocoon, and you are not running away or eating. So the faster you are done with that, the better. So it's a situation in which speed provides a clear advantage to the organism. Is also what we know about biologic systems are systems in which there is very high redundancy. There are different checks and balances to make sure that things are working appropriately. And one of the questions is, in which way are all of these things related? And so we looked at these questions using Drosophila, which is a workhorse for developmental biology. And the idea is that it goes through this life cycle, and this is where a lot of the complex developmental choices are made that give rise to all the different parts of the fly. And at each one of them, you have inputs that give signals, and you have ways in which you regulate those signals to make the appropriate proteins. And what we mean with there being redundancy is that there are usually a number of feedback loops that are controlling what's going on, because you don't want to have too much or too little or of some particular molecule. And when you do mutants in which you reduce the redundancy, usually that has dramatic consequences. And these are 3 examples of what would happen. Under normal development, you get these bristles here, always 3, always very nicely placed. If you make some removal in which you knock down one of the genes, what you find out is that instead of having 3, you have more, and some of the bristles don't look completely right. This is another type in which, again, you look at another difference. Instead of having these very, very nice 4 bristles in a very symmetric way without no problems and a lot of little bristles there, you have more in here, not symmetrically. The bristles look damaged. Or you can even have more dramatic stuff, like the eye that would develop in these pretty much perfect lattice becomes completely deformed and is unable to function. So we know that this redundancy under normal conditions is actually essential to have the correct development. Now, what we also know is that development depends on the metabolic rate, metabolic rate. So depending on your metabolism being normal or being slowed, and the metabolism can be slowed by affecting the way in which you process sugars, the time that it takes from you coming out from pupa to adult fly changes. And you can see that there is a dependence on temperature, but there is also, for all temperatures, when you slow the metabolism, you take longer to develop, meaning that you are longer in a sensitive state. Not only that, you are also smaller. So this is the small metabolism fly, and this is regular metabolism. So, but you can see that there are no imperfections. The process just takes longer, meaning that this is not advantageous to you, and you are smaller. And this is work with my colleague Rich Cartu, Neda Bagari. She's a faculty member too, and Sebastian is the student doing this work. And so what we thought is that when you suppress this redundancy, you are going to have negative outcomes. And those negative outcomes is because you are not able to bring the system to a desired state fast enough. And so if you have these are incorrect. If you have a redundant system, then oh no, this is correct. If it is non-redundant, this is the frequency of error. So you have much more developmental errors than in the other case, in which essentially the error rate becomes very low. But we ask the following question. Well, the system was adjusted to try to grow fast because there is advantage. So could it be that redundancy is only needed because of the need for speed too? Right? So what we asked is, okay, what if we remove the redundant feedback control, but actually simultaneously also decrease the metabolic rate? And our prediction is that under normal metabolism, when you remove some of the repressors, you can have much more mistakes. So a darker yellow here means that there are more mistakes occurring as opposed to the redundant case. But now, if you decrease the metabolic rate, or if you decrease by interfering with glucose uptake, or if you decrease the number of ribosomes in the organism, it actually also slows down your development because you are less able to produce proteins. What you find out is that under those repressed cases, you are actually able to dramatically reduce the rate at which errors occur. And actually, that's what you can find out. So this was from the model perspective. We can model this process and see, are these redundant circuits there actually to enable the organisms to accomplish these things under higher rates of development? And when we go and look at this experimentally, so the purple here is slow metabolism, okay? And this is the percent correct outcomes. So in the wild type, it doesn't matter if it is normal or slow metabolism. You get no mistakes. You now make a mutant in which you reduce redundancy. For normal metabolism, you only get the correct outcome, in this case, 25%. But when you slow metabolism, you get the correct outcome 75% of the time. This is another mutant, and again, you get a very, very significant difference just by slowing the metabolism. This one is for eye development. Again, if you are in the wild type condition, you get good outcomes no matter if it's slow or fast metabolism. You go to normal metabolism with the mutant, essentially you never get any eye development that is correct. You slow the metabolic rate, and 50% of the time, the eye develops without any problems. This is even a more dramatic expression in which you are essentially getting rid of all microRNAs. And you can see, again, that by slowing metabolism, you get the right outcome. So the idea, what this seems to suggest, is that there is actually a kind of virtuous cycle. If you are able to have greater redundancy in these networks, then you actually can develop faster. You can have all your cellular processes occurring faster, which means that you are in a sensitive, fragile state for less time. And this would give a driving force for creating greater redundancy and complexity of these gene regulation networks, because you get the benefit. You now can do things faster. You can work at higher temperatures. You can work at higher metabolic rates. You can be bigger and get out of this developmental stage faster, which gives you a big advantage. So we were very excited about this. Anyway, this is my last slide, I think. Yes. And I thank you very much for your attention, and I will be happy to answer any questions, if my brain is still working.
Thanks. From social stuff to great services.
Yeah, just because this attaches right to the last story, or the last slide actually you showed. So when you see that, because you started out by saying that you can slow metabolism in basically two ways, either by some kind of
yeah, you can play with the insulin pathway.
So you see these effects on redundancy both when you change slow metabolism by changing temperature, as well as when you actually take engineering.
Yeah, you can. The effect with temperature is very subtle, but you see a little bit of the effect. You can do. You can dramatically reduce the number of ribosomes that the cells have, and so this means that when you go from messenger RNA to protein, this is occurring much slower, and you can do it by messing up with the insulin pathway, and which again gives the organism has less ATP, and so it cannot grow, it cannot have the process occurring as fast.
You can get a similar reduction in speed of metabolism by changing the temperature enough, or by doing your sort of, engineer changes.
Mm-hmm.
But you're saying that the temperature effect is much more subtle? Because then it's basically, my question is basically around this, is it really just the rate of metabolism, or is it the whole mechanism of how you speed up metabolism that is more susceptible to, or that requires more error correction?
So the results were very comparable with the two ways, the ribosome reduction or the sort of ATP availability, that we could see the results. And in there, we actually did pretty brutal perturbations, right? We kind of, slower metabolism is slow by a factor of 2. With temperature, it's actually difficult to play with temperature that much that you can get that brutal an effect. Because at some point, the temperature is going to be so low that the organisms have trouble. But you know, it's something that we want to explore, and I think it would be nice. Well, we did these studies with Drosophila because that's Rich's organism, right? It's like, you know, where are we going to do this? We are going to do it on Drosophila, which is the organism that we know how to do everything. You could do these things maybe with bacteria, in which changing temperature could have a bigger effect on metabolic rates in there, but you know, as that was not the natural thing for him to do, and I was told that I should never enter an experimental lab, so I'm good with that. And
the question is one of hemostasis in the first case, where you're trying to gain temperature, because right. The next case is, as he has mentioned, you're actually doing something, a genetic mutation. You're actually introducing into the system.
yes.
Right. So my question is, I'm a little confused. Where exactly your redundancy comes into play?
So essentially what you do is you make a change when you are doing the small metabolism that you know leads to kind of a longer period for the developmental stage. And you also know that leads to smaller organisms. But you can see that the organisms look perfectly normal otherwise, right? So it's true that we are making more of a perturbation that you would expect, because you are actually making some genetic changes, but we believe that that perturbation is not very significant in all these other processes, except that it makes things kind of occur slower. Then what you go and do is, for instance, if you want to affect eye development, you go and do a mutation in which a particular microRNA, MER7, is knocked down. And so that is one of the feedback loops that MER7 is acting as a feedback loop to kind of regulate a particular process when during eye development. So you start with this cell of undifferentiated cells, with this disk of undifferentiated cells, and then they kind of develop in turn and become photoreceptors and cones and lenses and all of that stuff. And if you knock down this thing, then you see that the eye doesn't look normal at all. So at regular growth metabolic rates. And then when you look at the same thing, knocking, so the other case is going to have two mutations, right? It's going to have the mutation that changes the metabolic rate, and it's going to have the mutation that knocks out this MER7. And what you find out is that under these conditions, a lot of the eyes end up developing normally. And so it's sort of, you talk about rescuing the system from this. So it's the traditional thing in this, it's not you can just make one change for studying the effect of the knockout with the slower metabolic rate. You are making two changes, but sort of you have some confidence that the first change that just changes the metabolic rate doesn't have very much of an effect on things, because under normal conditions you see the organism developing normally.
A follow-up question on the question about temperature. It seems you briefly described the other ways that you slow the metabolism. I don't know much about the details here, but it sounds like in both of those other two, where you're messing with the ribosomes and with ATP or glucose uptake, you're sort of either introducing or perhaps modifying some bottleneck in your process. Whereas with temperature,
you would affect everything
you're
at
just
once.
sort of doing an overall, I think about like an Arrhenius law, you're just sort
Exactly.
of slowing down the overall reaction rate, but not changing kind of the degree of parallelism, if you will.
Yes.
Is it possible that that has something to do with why you see less of an effect with temperature, but more of an
Yes.
effect when you're sort of
I think that would also be one of the explanations, and we are not sure if it is because we are limited, in what range of temperatures we can really focus. In the model, when you put an Arrhenius law for the reactions and for the rates of everything, you really have to knock down to change the temperatures a lot to start to see that magnitude of effect that you saw in those diagrams. When you are looking at, kind of having the number of ribosomes, you saw already something very big, but it may be because that's a one bottleneck there, and it lowers the level of everything. So it would be nice we are thinking about looking at cell cycle which again is a process in which you have something that you have to be very careful doing while the cell is dividing and copying all the and again, with bacteria, with playing with temperatures, you kind of can have a bigger impact in there. But we are still waiting for that.
People working on data science. So your first two stories were about data science. Right now we're actually having a new institute here, data science. So data is basically easy to collect. Right. So what would be for you the challenge that we have? Like right now, you're trying to get patterns. So what is you're working with massive amount of data. So what is the?
So I see data science in, and this is something that I've been talking a lot to people. So I definitely like data science as a term much better than big data, because big data. People say, oh, only if you have petabytes or whatever and I always think, there are some people that are working on digital humanities, right? And it's sort of, instead of having one manuscript or something like that, now they have 1,000, right? And they could do something with one. They cannot do the same with 1,000, right? They cannot be reading each one of them and annotating all of them. So you need to move to another approach to look at data. And I think these challenges are kind of pervasive through many different areas. People are doing analysis of paintings, and there are all sorts of things that can be done once you start digitizing information in different contexts. I think that this kind of approach to create knowledge is very, very important. So for the longest time, we had two ways to create knowledge, right? You could be an empiricist, do experiments and have observations, or you could be, thinking of theories and deducting things. And Then we had computers that we could use to models, and then this was sort of an intermediate experimental system, right? We had more control over our computer experiments than we had in real experiments, because we really could determine what was in there, and we actually could look at things that maybe in a real experiment we couldn't. So that was cool. But a lot of the stuff that is going on, I think, with data science is a context in which you cannot make any experiment, right? I mean, hopefully you are not going to start a war or something like that, or again, hopefully not a civil war, right, with all the claims about rigged elections. But you cannot make experiments, but that does not mean that you cannot collect data and kind of find ways to extract quasi-experimental results by matching subjects and all of that stuff. So it's a very powerful way to learn stuff. And it's also incredibly powerful. For instance, I think in medicine, the applications of starting analyzing the electronic health records would be tremendous, right? It's sort of clinical trials with patients that are only taking one drug, and these and that and the other are so artificial that you learn much less than you would see what the effects are of people in the real world. So, and I mean, it's also great if you want to sell people things, and that is fully developed. But I think on the side of medicine or understanding violence and patterns of poverty and all of that stuff, the data that we are collecting could be used for us to learn a lot about the systems. It's a question of going and be able to use it, and for the people that understand the system to learn the tools that they need in order to be able to do that kind of analysis. So I think it's a tremendous opportunity for, so we right now have, for the school shootings, we have, I have a student that is co-advised. She's a sociologist and is going to be co-advised by me and the sociologist, right? And it's, it's cool because they are going to be learning about some techniques that they wouldn't otherwise, but they are also learning about all the sociological theory and things that you wouldn't know by yourself because you don't have time to go and learn the field. So I think it's very, very exciting, and